Hello everybody. We are in the second epidemiology session, and we, Donald King and I, will moderate this one. So Donald, back to you.
Yeah, thank you, Guido, and welcome, and a big hello to everyone. And for those of you who don't know me, my name is Donald King, and I'm head of the World Reference Laboratory for Foot and Mouth Disease, based at the Purbright Institute. And as Guido has already introduced himself, as the co-partner in crime today to chair this second session on epidemiology. And so the theme of today's talks will complement, I think, the first session from yesterday on epidemiology, and we'll focus here on molecular aspects of the virus and, if you like, the topic of molecular epidemiology. But before we go ahead with the talks, I've been asked to say a few words to introduce this session. And so we are all well aware that sequences are widely used to characterize FMD viruses that cause field outbreaks and help us understand the way that viral lineages are distributed in endemic settings. In this regard, the endemic pool concept that was introduced by Payton et al. In 2009, if I remember correctly, is a very useful tool where the 7 endemic pools that were described, with 3 located in Asia, 3 in Africa, and 1 in South America, help us to compartmentalize the global epidemiology of FMD. And these pools represent discrete ecosystems that are home to their own resident FMD lineages and maintain these lineages year on year. However, it's important to recognize that these pools are not completely closed systems. And when longer distance transport movements of viruses occur, these events come under close attention since they can pose new challenges for FMD control in these regions. So within and across these different endemic pools, there are complex dynamics that drive the dispersal of FMD viruses. And there have been a number of studies that have put forward the hypothesis that these virus movements are influenced by factors such as viral fitness, although we still don't really know how to properly measure this, as well as the existing immunity in hosts. And these virus movements are also shaped by other external factors, such as trade in animal and animal products, both regulated trade as well as non-regulated, perhaps illegal trade. An important factor that sometimes limits our ambition to understand the epidemiology of FMD is the fact that only a very small proportion of the outbreaks that occur in the field are actually sampled. And yesterday we were reminded about this so-called tip of the iceberg concept when it comes to sampling of field outbreaks. And there are perhaps new opportunities now to use sequence data and certainly sequence data diversity measures to estimate the numbers of animals that are actually infected with each of the FMD lineages. And in terms of the methods that are used for molecular characterization of FMD, there continues to be a focus on VP1 sequencing, but I think it's also accepted that full genome sequencing protocols are also well embedded in a number of laboratories that help to provide data that increases the resolution by which we can perform these analyses. Indeed, on Monday, during this conference, we've already heard about new sequencing methods that have been developed recently that may provide new opportunities to collect more data from the field. Taken together, a really important point is that, of course, sequence data is now becoming much more easier and cheaper to generate, and that new tools are also becoming available to use these data in an analytical context to address important questions relating to the epidemiology of FMD in endemic settings. But I suppose it's also really important to recognize that progress in this area is dependent on active partnerships between colleagues in the field that are collecting the materials, the laboratory scientists that are analyzing the samples, as well as the analytical computational scientists who do a lot of the clever work to make sense of these data. And so today, in today's session, we will hear some more about factors that drive the dispersal of FMD in East Africa, as well as 2 further talks that have analyzed FMD sequences in Pakistan and Argentina. And we will also hear about a new open access database, which we believe will help to facilitate the exchange and dissemination of viral sequences between the FMD community. So that brings to the end my opening presentation, and I pass the floor to you, Guido, to introduce the talks. Thank you.
Thank you very much. Thank you very much, Donald. And as we probably already know, we will first have the invited speaker, and then we will have our three speakers from selected works in this Congress. So our invited speaker is Anna Munse, now working in the Center for Disease Control and Prevention in the United States. And his work is phylogeographic analysis of foot and mouth disease virus serotype O, dispersal and associated drivers in East Africa. We will hear now his recording. Here.
Hi, today I'm presenting a phylogeographic analysis of foot and mouth disease serotype O dispersal and associated drivers in East Africa. This work is in collaboration with our colleagues at USDA Plum Island and Makerere University in Uganda. I'm going to be discussing two separate but related studies. The first study covers East Africa broadly. The second focuses specifically on Uganda. Uganda is the small landlocked country that sits on the northern border of Lake Victoria, and it's considered part of Pool 4, which is shown here with this red circle. Pool 4 has multiple circulating serotypes, OA, SATs 1, 2, and 3. And this area faces several barriers to foot and mouth disease control that include its high genetic diversity, frequent animal movements, and large populations of livestock and wildlife. Our main question for this study is, what are the drivers of serotype O transmission? We chose to focus on serotype O because it's the most common serotype in this area. And to do this, we utilized phylogeography to elucidate risk factors for spatial diffusion. We undertook sampling of cattle in Uganda over a 4-year period, including probang sampling, which is what's shown here. And our collaborators at Plum Island sequenced the VP1 coding region from viral isolates collected during probang sampling. For the first study, we combined Uganda sequences with GenBank sequences from across East Africa and utilized established phylogeographic tools, BEAST and Seraphim. And for the second study, we focused on Uganda sequences and developed novel regression tools. BEAST is a phylogenetic software package that incorporates a Bayesian approach. And the advantage of a Bayesian approach for this type of study is it allows us to integrate spatial temporal data into the tree. So our input for this BEAST analysis is the VP1 sequences with associated sampling dates and locations of the sampled animals. The output is our phylogenetic tree with dates and locations of the inferred nodes. And from this, we can estimate things like substitution rates, dispersal statistics, as well as test our hypothesized spatial risk factors. We started by combining our serotype O sequences collected during our Uganda study with GenBank sequences, all serotype O GenBank sequences collected from cattle in East Africa. So we started with this really large phylogenetic tree shown here, and then from there, we selected the monophyletic clade that represented sequences recently circulating in East Africa and did some subsampling to end up with a final data set of 148 serotype O sequences. And the phylogenetic tree produced by BEAST is shown here with the timescale shown here on the x-axis. This is the same phylogenetic tree shown in the last slide, now just color-coded by country of origin of the sequence. So you can see our Uganda sequences as well as GenBank Uganda sequences are colored in red, sequences from Ethiopia, light blue, sequences from Tanzania in green, and then sequences collected from Kenya are in this darker blue. The point of this is just to demonstrate the frequency of transboundary transmission during the period over which samples from this monophyletic clade were collected. this map shows the dispersal history of serotype O in East Africa. So it's essentially our beast phylogenetic tree in map format. So you can think of the lines as representing branches of the tree, the colored circles representing tree nodes. So the sampled nodes, or the tree tips, occur where a branch touches the earth and then stops, and then nodes that are connected by branches are inferred ancestral nodes whose locations and dates are estimated by the beast model. Shaded areas represent uncertainty of the inferred node, so essentially a spatial confidence interval, and then the nodes in shaded areas are colored according to their time of occurrence. The substitution rate for this analysis for the VP1 segment was 4.9 times 10 to the negative 3 substitutions per site per year, with the confidence interval shown here. For comparison, this is a higher substitution rate than both influenza A, which is the virus that commonly causes seasonal influenza, as well as for SARS-CoV-2. The reason we care about substitution rate for this type of study is that we know for rapidly evolving viruses like foot and mouth disease, the evolutionary and ecological dynamics occur on the same time scale. And this means that we can investigate the impact of environmental factors on virus spread. For the next portion of our analysis, we utilized a software package called Seraphim. And Seraphim extracts the spatial temporal information from the beast analysis, so from the phylogenetic tree. And what that means is that each phylogenetic branch can be treated as a movement vector associated with start and end locations and a dispersal duration. This is a kernel density plot showing variation in speed of spatial diffusion within the tree. So the spread upward and to the right indicates significant variation in mean diffusion. So some branches down here are less diffusive and likely represent local transmission among hosts, whereas other branches up here represent longer distance displacement. So likely something like human-mediated spread, movement of FMD-infected animals, for example. So because we have a high among branch variation and spread, we asked what are the drivers of this heterogeneity of spread? Using Seraphim, we can try to answer that question. So within Seraphim are methods to help us analyze the impact of environmental factors on dispersal, location, and direction. So we do this by calculating the environmental conditions at the locations of tree nodes, and then comparing those values to randomized branch positions within our study area. The first test analyzes dispersal location. So it looks at the statistic E, the tendency of lineages to remain circulating in certain environmental conditions. So it analyzes the mean environmental value at the node location and determines whether viral lineages have a tendency to remain in high environmental values or whether they have a tendency to remain in low environmental values. For this analysis, the tendency of lineages to remain in certain environmental conditions. On the left, you'll see our hypothesized environmental factors. On the right, the associated Bayes factors. And a Bayes factor greater than 3 is considered a positive association. A Bayes factor greater than 20 is considered a strong association. So our first hypothesized predictor was drought. So we used SPEI, which is a measure of drought. And there were no significant findings for this analysis. Log of cattle density, so higher cattle density was associated with the tendency for lineages to remain circulating in those areas. The same was true for population density, so areas of higher human population density, we found a tendency for serotype O lineages to remain circulating there. And then distance to livestock market, so longer distances were a negative driver, so indicating a tendency for lineages to remain circulating near livestock markets. The next analysis in Seraphim looks at dispersal direction. So using the statistic R, we're looking for a tendency of lineages to disperse toward certain lower or higher environmental values, so along environmental gradients. Statistically, it's the proportion of branches for which the environmental value at the oldest node is higher than it is at the youngest node. So do lineages tend to disperse from high values to low values, or vice versa, from low values to high values? For the statistic R, tendency of lineages to disperse along environmental gradients, we analyzed the same predictors and found no significant findings among those predictors. To summarize our findings so far, we found high variation in spatial diffusion of serotype O in this area, suggesting different modes of transmission occurring. For example, longer distance displacements suggestive of human-mediated spread in this area. For our location analysis within Seraphim, we found a tendency for serotype O lineages to remain circulating near livestock markets. That was the finding that had the most statistical support. We also found evidence of serotype O lineages remaining circulating in areas of high cattle density and high human population density. For our directional analysis, none of the predictors that we tested were statistically significant. So for the next analysis, we sought to develop tools which were more sensitive to directional trends or environmental gradients. For the next analysis, to develop our novel regression tools, we wanted to use a data set with relatively intensive sampling relative to the previous GenBank data set. So we focused on just our Uganda data, so a smaller area sampled over a shorter time period. And also we have a good understanding of the circumstances of sampling of these animals. This map is showing the geographic locations within Uganda of the 48 herds from which serotype O isolates were collected over about a 1-year period of our study. Once again, we used BEAST to construct a Bayesian phylogenetic tree, and we utilized Seraphim just to extract the spatial temporal information from the phylogenetic tree. This is just a video showing the dispersal history of serotype O during the period studied here. The first method that we utilized to assess directional trends is called a step selection function model. So step selection functions were originally designed for wildlife ecology to analyze wildlife habitat selection. So they correlate environmental data to animal location data by comparing areas that are used by the animals to locations that were unused but assumed to be available to them. So when we apply this to a viral phylogeny, we compare conditions at the branch ends, so the used locations labeled here with a black dot, relative to the available but unused locations designated with a white dot. And we asked, what is the difference between where the virus traveled relative to where it could have traveled under the same space-time constraints? The next tool that we developed is called a resource gradient function model. If you recall, the step selection function model utilized a matched used available design. The resource gradient function model utilizes a matched start end design. So this is comparing conditions at or near the branch start locations, shown by the black dots, to conditions at or near the branch end locations, shown by the white dots. In order to increase the statistical power of this analysis, we generated these neighbor locations for each observed location. And the point of this is to understand the gradient along which FMD lineages are tending to move so that we can better understand FMD source sink dynamics. This is a diagram depicting viral source and sink locations as applied to influenza. A viral source can be thought of as a sort of reservoir population. It's areas in which continual viral transmission results in greater genetic and antigenic diversity. So for influenza, it's been shown that the tropics serve as a viral source because in these areas a favorable climate allows for year-round transmission, resulting in higher genetic diversity shown by the different colors before being exported to the northern and southern hemispheres, which serve as influenza sinks. So source areas are areas in which control measures can potentially have a greater impact. For these novel regression analyses, we analyzed many of the same predictors that we did for the initial analyses: rainfall, cattle density, human population density. We also added distance to roadways and distance to international borders, since these predictors were informative in our earlier serology studies. What we found using the step selection function model was a marginal association with areas of low rainfall. So we found that locations in areas of low rainfall were marginally more likely to be classified as a used location as opposed to an available but unused location for the viral phylogeny. For our resource gradient function model, only the predictors that were significant are listed here. So rainfall, cattle density, and proximity to livestock markets. So areas of low rainfall were protective against being classified as a branch start location, indicating that they were more likely to be classified as a branch end location. Further, high cattle density in areas near livestock markets were more likely to be classified as a branch start location. Utilizing the results from our resource gradient function model, we assembled this map that depicts the qualitative risk of an area being classified as a source of FMD serotype O based on this analysis. So FMD in this case is likely to disperse along the red gradient, where higher values represent a higher likelihood of being a source of FMD, so classified as a branch start location under this model. Higher values represent areas with high cattle density, areas that are close to livestock markets, and lower values represent areas of lower cattle density, further from livestock markets, and areas of low rainfall. To summarize all these findings, we found evidence of significant transboundary transmission in East Africa, evidence of longer distance spatial diffusion, so likely anthropogenic transmission, as well as the following risk factors. So livestock markets, high cattle density are more likely to serve as FMD source areas, whereas areas of low rainfall, we found a tendency for virus to remain circulating there, but they were more likely to be classified as sink areas, so branch end locations in our resource gradient function analysis. And then pastoralism in areas near international borders were not significant. So how can this help guide outbreak response? So our hope is that, for example, if we have an outbreak that's starting in an area of high cattle density and close to livestock markets, that may encourage responders to kind of dial up their outbreak response. I've gone over time by a minute, so I'll quickly acknowledge collaborators and former advisors at the University of Minnesota, Kim Vanderwall, Andres Perez, Frank Monet and Silver Ochoa at Macquarie University, and then our collaborators in Brussels, Plum Island, Animal Disease Center, USDA, South Africa are all listed here, and our funding is listed here on the left. Thanks, I'm happy to take any questions.
We will go next with the three speaker presentation. So the first one will be Nick Knowles from the Purebred Institute in the United Kingdom, a new expert-curated foot and mouth disease virus nucleotide sequence database. The second one will be Syed Shamal from the University of Malakand in Pakistan, and the name of his work it is characterization of serotype Asia 1 FMDB circulating in Pakistan during 2012 and 2020, and identification of a new genetic group. Finally, our speaker will be Laura Camila Lozano Calderón. She is from the Instituto de Agrobiotecnología y Biología Molecular, INTA CONICET, Argentina, and the name of her work, it will be Evolution, Analysis, and Phylogeographic Reconstruction of Foot and Mouth Disease in Mar Chiquita, Argentina.
Curated foot and mouth disease virus nucleotide sequence database. The accessibility of foot and mouth disease genome data is either limited to personal data or data available in public repositories, with often non-existent, limited or incorrect metadata annotation. Third-party annotation of sequences on public databases is problematic, and inaccurate data is usually not corrected, which can lead to incorrect spatial or temporal phylogeographical inferences. The FAO World Reference Laboratory for Foot and Mouth Disease possesses a significant amount of unpublished sequence data, which are generally well curated. To address these issues, we have built an FMD network laboratory sequence database, simply known as FMD-Base. An open source relational database management system has been developed in MySQL in conjunction with Lightflows Limited. The database has been populated with FMD sequence data retrieved from 3 sources: GenBank, EMBL, DDBJ, the WRL database of unpublished VP1 sequences, and unpublished full genome sequences determined by WRL FMD. Data are being manually checked for inconsistencies and curated with all their required information where this is available. The schematic plan of the system is as such, and presumably mainly the database on the left hand side is what we're talking about today. There are further developments in progress to add tools to this database. The database has a number of entities, including sequence data, genomes, virus samples, and batches. Sequences belong to a genome, and genomes may belong to a sample. So there may be multiple sequences of a single genome, and there may be multiple genomes in a single sample. And then there are additional entities of taxonomic groups, countries, world regions, host species, and laboratories. And three security levels: public, private, and confidential. The data initially consists of GenBank data of over 10,000 records, of which just over 1,000 were complete or near complete genomes. And this will be added to by over 900 unpublished whole genome sequences and over 6,000 complete or partial VP1 sequences, including some submitted by other laboratories. So the total number of sequences added to FMD-Base so far is 16,764. This will increase over the next few months as more sequences are prepared for release. And this illustration here shows the main interface whereby people can access this data. The data can be searched and exported, and the main search filters are virus lineage, country, host, collection date, and genome region. And these can be used to search for a subset of sequences which can then be exported in FASTA format, or including with metadata in CSV format. So the release of the database for external access is expected by the end of November 2021, and the website will be www.fmdbase.org. The accessibility of the system for researchers will initially be via a web portal and based on a three-tier security structure, allowing different users access and retrieve data. Public data will be accessible by everybody, but private data, including unpublished sequences from WRL partners, will be authenticated and accessed via an agreement in terms of an MOU. In the future, we will like to develop online applications for dynamic data query and visualizations, and a harmonized repository of FMD genetic and epidemiological data. We're in the process of developing front-end dashboards with the New Zealand company Epi Interactive, headed by Drs. Petra and Uli Mulner. One of these dashboards will be an FMD genotyping system, which will produce a report which includes a phylogenetic tree, and this will be available to all users for their own data. So remember to look out for this web address, www.fmdbase.org, and announcements that it has been released, hopefully in the near future. So the acknowledgements are shown here, and this work has been partially paid for by the UKRI Global Challenges Research Fund, and the WRL is supporting this sequencing by various others, including DEFRA UK. Thank you.
Hello everyone, good morning and good evening wherever you are. My name is Sayyid Jamal and I am from the Department of Biotechnology, University of Malakand in Pakistan, and the title of my presentation is Characterization of Serotype Asia-1 foot and mouth disease viruses in Pakistan during 2012 to 2020 and detection of new genetic group. Foot and mouth disease is endemic in Pakistan and 3 serotypes of the virus are involved in disease outbreaks. The serotypes are serotype O, A, and Asia-1, and these belong to pool 3 viruses. However, there has been detection of serotype O belonging to pool 2 virus in Pakistan in 2019. Multiple subtypes within each serotypes are involved in disease outbreaks in the country, and new subtypes within each serotype arise with time. The rate of evolution of serotype O indicates that it takes 5 years to develop a new sublineage within O-Pan Asia2 sublineage. However, the rate of evolution is high in AIRAN05 lineage as compared to O-Pan Asia2, and thus the new subtypes or new sublineage within AIRAN05 lineage arise within 3 years period. There is circulation of diverse subtypes within the country and even within the same serotype. Circulation of foot and mouth disease viruses is important. Characterization of foot and mouth disease virus is important for not only for the selection of appropriate vaccine but also for tracing the origin of outbreaks. There has been 9 different genetic groups within Asia-1 which have been recognized so far in Asia, and out of these 3 genetic groups, that is group 2, group 6 and group 7 of serotype Asia-1 have been found circulating in Pakistan. The objective of this study is to characterize serotype Asia-1 foot and mouth disease viruses circulating in Pakistan during 2012 to 2020. In this study, samples were collected from clinically suspected cases of foot and mouth disease viruses, and these samples were tested using antigen ELISA, and samples which were tested positive using antigen ELISA were further tested using RT-PCR to amplify the VP1 coding region, and the complete coding nucleotide sequences were generated from the amplified product, and then phylogenetic tree was constructed using the VP1 coding sequences from these viruses and also from some reference and old viruses which are present in the public databases. Phylogenetic analysis revealed the presence of 2 different genetic groups of serotype Asia-1 foot and mouth disease viruses in Pakistan during 2012 to 2020, and these 2 groups included one which has been previously recognized in Pakistan and has been named as group 7 or SIN08, and there has been detection of a new genetic group which is designated here as group group 10. It is to mention that group 8 and group 9 have been detected in Bangladesh during the previous years. The group 7 or SIN08 group was first detected in Pakistan in late 2008, and it was later found in Iran and in Afghanistan and also in the Middle East, and which reached to Turkey in 2010 and 11. And the highlighted portion represents the group 7 viruses in the phylogenetic tree. It was last detected in Pakistan in 2019, and this genetic group has not been detected in samples collected in Pakistan during 2020. The second genetic group, which has been detected recently, and which is new genetic group and which has been designated as group 7, it was first detected in samples collected in Pakistan in early 2019. And the viruses belonging to this genetic group has been found circulating in at least 2 provinces in Pakistan, that is the eastern part of the country, that is Punjab province, and northwestern part of the country, that is Khyber Pakhtunkhwa, during 2019 and 2020. In 2019, there has been detection of both the genetic groups, both group 7 and group 10. However, in 2020, only group 10 has been detected in samples from Pakistan. So, from this study, it is concluded that foot and mouth disease viruses circulating in this region are continuously evolving, as revealed by the detection of a new genetic group, designated here as groups 10. This study motivates continued FMD surveillance to monitor the circulating FMDV subtypes and to improve our understanding of the epidemiological risk in the region. Further studies are required to ascertain whether anti-sera raised against the available vaccines are able to efficiently neutralize viruses belonging to this new genetic group, or whether a new vaccine strain is required to be used for the control of this new genetic group. We would like to thank Salman Khan and Mohammad Isaq from the Department of Biotechnology, University of Malakand, Dr. Syed Asad Ali Shah from Livestock and Dairy Development Department, Khyber Pakhtunkhwa, and Dr. Hanifur Rahman from Veterinary Research Institute, Peshawar, and also Graham Bilsham from the University of Copenhagen, and Michael from FLI, Reims, during the course of this study. Thank you so much.
Good morning. It's a pleasure for me to be here. So today I will show you this work that I released, Evolution Analysis of Foot and Mouth Disease in Marchiquita, Argentina. This is only a quick introduction. As you know, foot and mouth disease is a highly contagious disease produced by apicornavirus, single chain, member of the aftovirus family. It affects club of animals and have an important economic impact. Well, about epidemiology, I will say that the disease is distributed in many countries. However, Argentina now is free. But in 2001, the country had a very important epidemic that left data, and on some of this study is based. Well, so the specific objective of this study is analyze the viral evolution in an Argentina district and propose possible transmission routes. But why this is important now, 20 years later? So we believe that it's very important keep one system ready for a possible new epidemic. So we work in models that in the future could be adapted. Well, the methodology was the following. First, we select samples with traceable epidemiology data, and later the samples were inactivated with trisol. Subsequently, we extract the genetic material and made the synthesis of cDNA. Later, all genome was amplified by PCR and sequenced by Sanger technique. After that, we aligned and cured the sequences. We estimate also the evolution models and build phylogenetic tree. These phylogenetic trees were built through maximum likelihood using IQ3 program. And finally, the evolutionary analysis and phylogeographic reconstruction were carried out through these programs. We visualized the running using tracer, and the transmission routes were shown on the map using Sprint and Google Earth. So we were the following three. Here we can observe the grouping of three clusters, 1, 2, and 3. And this is an outgroup. We use the samples called General Lopez. Well, about the evolution analysis, we can observe that the population size grew gradually. After the dates analyzed, we don't know the population group. The viral evolutionary rate was 9-15 times 10 to the negative 3. We can find also that the most recent common ancestor diverged in April, 1 month before. So only for be clear about this running, we reach convergence running 10 million generations. Okay, in this slide, I will show you the viral transmission animation. In this point, we couldn't find a good correlation between geographic and genetic distance. We can find also that in this data set, the majority of gene samples are located in the center and north of the district. So finally, we can conclude that in Mar Chiquita district, there are evidence of at least 3 independent transmission routes, and was also possible identify relative long geographic links between pairs of very similar sequences. This suggests that the different spread routes as no replication vectors like water, human transport. The viral evolution rate was 950 times to the negative 3, and all the samples sequences diverge for the most recent common ancestor 1 month before first event report. Yes. So thank you so much for the time. And I'm going to be able to answer questions. You can also send us an email. This is all. Thank you so much for the time.
Thanks for those great talks, and welcome now to the discussion session. So the talks are now open for questions from the audience, and we will try to, Guido and myself will try to pick up as many of the questions from the chat box as possible. And so feel free to type in questions relating to the talks that you've heard today. And so Guido, do you want to kick off with the first question?
Yes. This is the first one is for Anna, and you identify a low number of outbreaks in Uganda. How many epidemiological units are there in Uganda? In another word, how many infected farms did you miss, and how would that influence your analysis?
That's a good question. So I don't know the number of epidemiological units. The last estimate I saw in terms of number of cattle was around 10,000 in the country, and the herd sizes vary really widely from very small, 1 to 2 animals at a household, all the way up to very large farms of thousands of animals. So if I had to guess, I would say the number of epidemiological units is probably in the thousands, low thousands. How many did we miss? That's a good question. And I wish I knew. I think outbreak reporting in Uganda is not quite complete, so it's hard to know. But we don't have a reason to believe, I guess, that the herds that we included are at least not representative, or we believe they're representative of at least the serotype O sequences that were circulating at the time. So.
Thank you. So can I ask another question to you, Anna?
Sure.
I mean,
Yeah.
while we're on the topic of your presentation. So, I mean, I think that a term that's widely used now to sort of describe endemic FMD circulation is this concept of the source sink model. And I'm just sort of questioning perhaps or challenging that idea. Is that really a model that has immediate relevance for the way that FMD is circulating in endemic settings?
I think it partly depends on the control measures in place. So in Uganda, our hope was understanding something like this would be useful because they use this kind of like fire brigade response to outbreaks where they come in with vaccines after an outbreak has been reported, and often the outbreak has been circulating for some time. And so our hope is that if we understand, because we have a limited number of vaccines available to use in Uganda and they're vaccinating during ongoing outbreaks, we hope that they can kind of direct those vaccines, those limited number of vaccines, to the areas that are more likely to serve as viral source areas. So the areas that are more likely to serve as areas of high transmission. In terms of other endemic settings, I think it depends on perhaps the control measures that are being used there. I'm just most familiar with Uganda, so.
Thank you. We'll probably pick up some more questions for you in a minute,
Yes.
but perhaps if we can move through the rest of the speakers. Guido, do you want to ask Nick a question?
Yes. There will be a way to know how much of the data we are getting on the public side of the FMDB base versus how much more we could get on the mode side for a particular query?
The only way at the moment would be to actually register on the site, and then you'll be able to see all the data. We can give you an idea about how many sequences in total, but that wouldn't necessarily tell you, for example, if you were searching for a particular lineage, on how many were missing. So I think that might be quite difficult to do. Maybe we'll think about looking at the whatever's on the database and trying maybe to list it in some way so that people can know that. One thing I would say though, the database is now online. When I prepared the talk last week, it was not ready, but now it is. We are still testing out the registration process, so please be patient. But you certainly should be able to see the public data now.
And perhaps just to add to what you've said, Nick, I mean, the ambition here is to make all the data as available as possible. But as we go forward, we're imagining that quite a lot of unpublished data will be in, if you like, this private region. And so in order to access that, we make a very simple registration form that people fill in, and hopefully that will be acceptable by the community as a way of protecting the data at the same time as making it as available as possible.
Yeah.
So Najenda, I think, asks a sort of somewhat related question, which is will the sequences generated by WRL be public or private?
They would normally be private until we publish them. But I think in some cases in the past, for example, where an important outbreak has occurred, we've tried to submit data to GenBank, for example, very quickly, even before we published. So it will somewhat depend on the data. But of course, that private data will be accessible by network partners.
Okay. Thank you, Nick.
Okay.
Over to you, Guido.
Yes, we have one for Sayed. Do you know more about the new lineage of ACO1, such as virulence, and do you think this may be a new threat to the region? I think it's Sayed. We lost him.
I think you're on mute, Syed. Thank you.
Hello, Guido. Can you please repeat your question, please?
Yes, of course. Do you know more about the new lineage of ACO1, such as virulence, and do you think this may be a new threat to the region?
Well, actually, as far as lineage, I think you were asking about lineage, the new lineage 10 that we have designated. Actually, it seems that it is the new introduction in this region, but from where? This is the question. Maybe the virus has evolved, but as far as the nucleotide sequences are concerned, we have no clue. The nucleotide sequences don't provide us any clue to ascertain whether it has been evolved from group 7 or group 4 or any other group. So this is uncertain from the existing data.
Can I ask a follow-on question, which is particularly relating to Asia 1? I mean, I think we heard yesterday about the fact that Asia 1 is now disappeared from Southeast Asia as a lineage that is widely circulating, and so they don't have it present in that region. And I mean, we also heard yesterday a lot of discussion about serotype C, and so the obvious question is why is Asia 1 seem to have such a more restricted and limited distribution compared to some of the other serotypes? And can that tell us something about the risks and threats and perhaps the future for this serotype?
It's difficult to answer this question. I think about the restriction of serotype Asia 1 only to Asia, and similarly the restriction of serotype sets to Africa. But previously, serotype Asia 1 has been found very conserved, and the existing vaccine strains were able to contain the spread of this virus. However, as far as this the group 7 or SIN08 is concerned, the Asia 1 Shamir was not able to contain, efficiently neutralize the SIN08 or group 7 viruses of serotype Asia 1. And perhaps it's due to this reason that in 2008, when it was first identified in Pakistan in late 2008, it spread to other parts of Asia and reached Turkey in 2010. And it was responsible for huge outbreaks till 2019, at least in Pakistan. Previously, these were very conserved, but but endogenically not very much. There was no much difference endogenically. But the Asia 1 Group 7 or SIN08, it was endogenically different, and we don't know about this new virus, whether the new lineage, whether it is endogenically different or not. And I'm not sure whether you have received samples from MICA, from FLI, for endogenic characterization. If not, then I hope that this will be reached to you in near future. So we'll be able to know whether the available vaccine sera are able to neutralize efficiently this new lineage or not. As far as C is concerned, so it is still as far as the spread of the Asia 1 is concerned, it may be disappearing from other parts of Asia, but as far as Pakistan, Iran, Afghanistan, and this region is concerned, there has been continuous outbreaks of the disease due to serotype Asia. So maybe this may be a new next candidate, but effective vaccination and effective vaccine and potent vaccine may be required to get rid of this.
Yeah, thank you for those comments. Guido, do we have some questions for Laura?
Yes. How do you explain such a big mutation rate with FMD, which is around 6, 7 by 10 a la minus 3 by minus 3 compared to influenza A and SARS-CoV, which is a little bit lower?
Well, it's unusual to have a high rate with samples taken and in short time, but maybe we can think that in this moment these animals have a pressure for vaccinate. No, some animals were vaccinated.
Thank you.
So can I ask Laura a follow-on question for you, which I think comes from the chat box? which is from Rebecca. She says, I seem to remember prior analyses from the Argentina 2001 outbreak looking at pigs versus cattle. And so she's asking, were you able to consider the impact of or the influence of species in your analyses?
Now, no, only cattle.
So all the sequence data that you presented today is cattle only. Is that correct?
Yes,
Yeah.
yes,
Okay.
it's only cattle.
And just remind me, to what extent were pigs involved? I have to remind me and probably most of the audience too.
It's very, very, sorry, I will answer that it was very low. The incidence was very high in bovine herd, but the role of the pigs in the transmission of the virus was unimportant.
Okay, thank you.
So one last one for Anna, and then we finish. I think we are on time now. Anna, can you comment on difference in the source sink risk factor and that may be observed over a larger scale?
That's an interesting question. So we chose to focus on, for our study, this really small area of Uganda over a short time period, because this was kind of a proof of concept, us developing these novel regression tools. We wanted to have good confidence that all of the outbreaks were sampled during our time period and over this study area. So I think that moving forward, it would be really cool to expand it to a larger geographic area, but we haven't done that quite yet. So it would be interesting to try.
Well, excellent. Thank you very much all for listening, and to our speakers. Thank you very much. It was a great session for me.
And thank you also from me. Thank you to again for the great presentations. I'm sure if we were in a real format, everybody would be cheering and clapping the lovely presentations. And I thank you for the opportunity to answer, for taking the time to answer some of the questions. I can see that there are also some more questions in the chat box, and perhaps other colleagues have other questions. And so I'm sure that the speakers here will now decant into our virtual avatar format, and we can go into Gathertown for further discussions on these presentations. And so, Guido, thank you very much for helping me today, and thanks everyone for joining the session.
Thank you so much.